time, an irreversible force of nature, a merciless one-dimensional arrow to the future. We all have experienced it, are experiencing it, and will experience it. It is the key agent in the inevitable process of aging, an entropy-driven process that slowly weakens the body and slowing its function, eventually leading to death. It's quite macabre, isn't it? As the famous writer Beryl Bainbridge once said, "Nothing is so sad as the injustices of old age. No one chooses to age or to grow old. It's inevitable." Or is it? Let's define aging. Aging is defined as an inevitable time-dependent decline in physiological integrity and function caused by the accumulation of cellular damage, which drives a progressive loss in biological function. There are 9 types of cellular damage that are hallmarks of aging, which are all interrelated, affecting and in some cases exacerbating one another. However, we are going to focus on one important factor that affects the rest: epigenetic alteration. What is epigenetics? It is the study of changes in organisms caused by modifications of gene expressions rather than alterations to the genetic code itself. It plays a vital role in cell specialization by switching genes on and off in different cells in the organism, resulting in cells with different functions like nerve cells and pancreatic beta cells. These are various methods to modify the epigenome, like methylation, which is the addition of a methyl group to the C5 position of cytosine, and it represses gene expression. Histone acetylation, on the other hand, removes positive charges from the histone, reducing its affinity to the DNA. Ergo, the DNA is less tightly bound and transcription is enhanced. Due to specialization, certain cells are supposed to have certain epigenetic patterns, so they express the right genes, transcribe and translate the right proteins for its cellular function. However, over time, epigenetic noise throughout cell division and other factors like your diet, lifestyle habits, and environmental pollutants can change the methylation and acetylation patterns. This is measured by the Haworth clock that measures the deviation of described CpG islands methylation patterns from youthful methylation patterns in humans, which determines your epigenetic age. Changes in gene expression can disturb proteostasis, the balance of proteins produced, and the structure of proteins, affecting metabolic pathways that can lead to diseases like Parkinson's and Alzheimer's. If genes are overexpressed, they can form cancerous tumors that proliferate in the body. If genes are not sufficiently expressed, emergent functions can be affected, like age-related glaucoma, which is damage to the optic nerve due to epigenetic noise. This shows the significance of the epigenome in cells and the organism as a whole. What if there was a way to reverse Haworth's clock and return to more youthful epigenetic patterns? Shinya Yamanaka is an innovative Japanese scientist that worked on inducing pluripotency in human adult fibroblasts. Pluripotency means, so pluri means many, and potency means capacity. So it's the capacity of a cell to initiate all lineages of a mature organism. Yamanaka and Takahashi first discovered 24 potential reprogramming factors that were abundant in human embryonic stem cells and injected them into mouse skin cells. They discovered that none of them were able to induce pluripotency. However, after vigorous testing, they found out that a peculiar cocktail of 4 proteins, OCT3/4, SOX2, KLF4, and CMYC, was able to induce pluripotency in mouse skin cells. These forgettable names are transcription factors which control the rate of transcription of certain regions of coding DNA from DNA to mRNA. The Yamanaka factors bind to promoter sites in the cell genome, allowing RNA polymerase to transcribe select regions of DNA into mRNA that is further translated into proteins that sends signaling pathways through the cell that changes methylation patterns back to a more youthful state. If the Yamanaka factors are introduced for a long period of time, the cell will lose all methylation patterns of the DNA, returning to a completely unspecialized state as a pluripotent stem cell. This is referred to as an induced pluripotent stem cell, or iPSC for short. Creating iPSCs in the body is dangerous, forming teratomas, which are tumors that contain immature and specialized cells, including nails, teeth, muscle, and hair. Hence, scientists building upon Yamanaka's research chose to work with Yamanaka factors that can be switched on and off. In the experiments below, mice were injected with viral vectors with the Yamanaka factors and a TET-off promoter that detects tetracycline and its derivatives like doxycycline, or DOX for short. Hence, the mice will only produce the Yamanaka factors when fed with DOX-filled water. Belmont's team works on mice suffering from progeria, a progressive genetic disorder that causes organisms to age rapidly. The epigenetic marks on DNA are dysregulated in mice and humans with progeria, so the team used progeria mice skin cells genetically engineered with Yamanaka factors and fed them water with DOX for a short period of time. The animals' lifespan was increased by 30%. They looked younger and cardiovascular functions improved. Damaged muscles and their pancreas were also rejuvenated using the same method. Not long after, David Sinclair, Rencheng Liu et al. Restored vision in old mice and glaucomic mice using the same method. Viral vectors with TET-off promoter genes that transcribe and translate into Yamanaka factors were introduced into these mice optic nerve cells. The mice were fed DOX-filled water for a few days, and miraculously, the optic nerve cells regenerated, improving visual acuity. This was surprising as mammal central nervous system becomes senescent early in development. This definitively showed that the biological clock was reversed. This is just the beginning of the story. There is a reason why Shinya Yamanaka won the Nobel Prize in Physiology and Medicine of 2012. Regenerative medicine is the future, and in vivo epigenetic restoration plays a vital role in it. It is non-invasive, recovery time is minimal, and no foreign cells are introduced. The feasibility of the project makes it even more alluring. The cost of an iPS reprogramming kit at commercial price is around $1,000, which will drop over time as this technology becomes more established. It is readily available by multiple companies as it was not patented, and it is environmentally friendlier than surgery or chemotherapy. It has the potential to cure myriads of diseases, starting experiments in mice and trickling up to actual patients suffering from these diseases, including diabetes, various cancers, heart disease, and even cosmetic applications like making one's skin look younger or healing permanent skin wounds. Our body has a clock that predetermines our fate, our bodily function, and our health. Or at least it used to.